Good morning to all.
Good morning, ma'am.
Good morning, students.
Good morning.
Good morning.
My sound is audible.
Yes, ma'am.
Yes, ma'am.
Okay. Shall I start the practical today?
Okay, ma'am.
Okay. 69 participants are there. What about others? What about them? Some newcomers also there now. Web.
Yes, miss.
So what about others?
Totally only 70 participants in our group, miss.
70.
70.
Okay. Okay. So today we will look the gel electrophoresis. Okay. In your first batch, they studied about the preservation techniques, isn't it? Did you heard that one? Did you heard? What's the last practical of your other batch? Is my sound is audible or not?
Yes, ma'am. Audible.
Okay. If you respond only, I can know whether it is clear or not. So, did you discuss your other batch regarding the lab techniques? Any one of you?
Yes, ma'am. They say the last class preservation of specimens.
Yeah, they studied the preservation of
Specimens.
specimens. So, in your upcoming class, you may study that one, okay? Today, you will look the gel electrophoresis. In your upcoming, I think the Thursday, you may study the preservation techniques, okay? So, don't worry about that one. Today, we will look the gel electrophoresis. Anything do you know about this gel electrophoresis? Did you hear the word before? Gel electrophoresis?
Yes, ma'am.
Yes. Do you know about this one?
The method of arrangement of short DNA fragments.
Yeah, good. So, the gel electrophoresis. Okay. So, the technique that is used to separate the biological molecules under the gel based on the size and charges. In case of the gel electrophoresis, where we can separate the biological molecules like DNA, RNA, proteins, amino acids, or we can separate under the gel based on their size, shape, and their charges. Okay, it is achieved by the loading them in a gel and creating an electric field across the gel. In case of the DNA, protein, RNA, amino acid, we should load in the gel and we should apply the gel electrophoresis. In here, the gel that is act as a molecular seal. The gel that may act as the molecular seal. We can use the gel, agarose, polyacrylamide amide, agar, and starch. There are different types of gel we can use in the gel electrophoresis. The some of the examples are agarose, agar, polyacrylamide amide, and starch. Okay, these are the gel we can use in the gel electrophoresis. In this diagram, you can observe the agarose and polyacrylamide amide structure in this picture. Okay, understood?
Yes, ma'am.
Okay. So then, what's the gel electrophoresis? What is it? What's that one?
Separating the biological molecules according to their shapes, size.
Sorry?
Separating the biological molecules according to their shapes and size.
What are the biological molecule you can separate?
DNA, RNA, protein.
DNA, RNA, proteins, amino acids, and some of the macromolecules. Okay. In here, the electro means electricity. Okay. What's that? Electricity. Phoresis means separation. Gel, that is the semi-solid medium. Can you visualize this one? This slide? Can you?
Yes, ma'am.
Okay. So in case of the gel electrophoresis, from this one you can make the definition. Separate the macromolecules under the electric field in a semi-solid medium. That is the gel electrophoresis. In here, the separation that may be made out based on the size, shape, and the charges. Okay? Okay. So in here, the molecular movements also occur. Isn't it? The molecules are moved in this semi-solid medium or gel. Yes or no?
Yes, ma'am.
No. Yes or no?
Yes, ma'am.
Yes, ma'am.
Okay. So that in this electrophoresis, we can tell electrokinetic phenomenon. Electrokinetic phenomenon. Because in here the molecules are moved under the electricity or electric field. That's why in this phenomenon we can say the electrokinetic. Okay. So in here the molecules are moved. So what do you think about that one? How it is moved? We can make any pressure or artificial inserting anything. How the molecules are moved? Any guesses? Why all of you are in silent mode?
Due to the net flow of ions or electrons. And,
Yeah, good. Due to the charges, net flow of electrons. In here, we will external electric field. Okay. So due to that one, if the molecules have the charges only, they can respond under the electric field. Okay. In here, what are the molecules we can separate it? Yeah,
Amino acid.
RNA, proteins, and amino acid. So that they should have the charges. Then only they can move through this feed, isn't it?
Yes, ma'am.
Yes. If you actively participate only, I can tell more. I also in the active mood. Okay. If you are in the dormant stage, then I can't. Okay. Please
Okay,
respond
ma'am.
as soon as possible.
Okay.
Okay. So, in case of the DNA, RNA, protein, amino acid, we can separate it by using this gel electrophoresis. So, if these macromolecules, if you want to respond, they should have the net charges. Okay, that's why in here the DNA and RNA. DNA and RNA. What do you think about the charges? The DNA, RNA charges, whether it is positive or negative. What do you think about that one? In case of the DNA, RNA, these are negatively charged due to the presence of phosphate group. Okay? Due to the presence of phosphate group, it is negatively charged. And the protein may be positive or negatively charged. It is depend on the pH. Okay, protein that may be positive or negatively charged, that is depend on the pH range. But the DNA, RNA which are negatively charged. Okay, so due to this presence of charge, they can move across this gel under the electric field. So what do you think about that one? Negatively charged one, sorry, negatively charged one can go towards the positively charged, isn't it?
Yes, ma'am. Yes, ma'am.
Yes, the negatively charged molecule that can be attract by the positively charged node. Okay, in case of the positively charged molecule, that will be attract to negatively charged node. Yes or no?
Yes, ma'am.
Yes, the negatively charged molecules may be the anion that will be attacked towards the positively charged node. The node that we can say the anode. Okay, negatively charged molecules, anion that is attacked to positively charged node. That is the anode. So what do you think about that? Positively charged molecules. After your response only, I can go further.
Cation and cathode.
Yeah, positively charged molecules that we can say the cations that will be go towards the negatively charged node. That is the cathode. Okay. Did you understood the gel electrophoresis basic?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Any one of your batch, can you tell the short summary of this one?
Ma'am,
Yes. Yes, you can say. Takshila. Web. Representative, are you there in these lectures?
Shall I do?
Ma'am, I can summarize.
Representatives, are you there?
yes,
Okay,
miss.
can you tell about the gel electrophoresis until now what we discussed? Why you are so lazy?
And can I explain?
Yeah, you can.
Gel electrophoresis is the method to arrange to separate the small according to the size of the molecules DNA or RNA. And this method, the electricity is passed away the gel electrophoresis plate, and that's why the molecules are separated according to the sizes. The negative molecules are goes to anode, and uh, positive molecules are attacked towards the cathode.
Yeah, good. So then these gels, what are the gels we can use in this electrophoresis?
Agar, starch, polyacrylamide.
Agar, agarose, starch, polyacrylamide amide. We can use as a gel. In here, the gel that is act as a molecular sieve. Okay. In the earlier time, in this structure, assume the agarose. In here, in this agarose, that is act as a molecular structure. Molecular sieve. So the molecular sieve means that can only allow smaller diameter. Okay. This is the molecules that can allow only the smaller diameter than this one, this pose. Okay. Smaller diameter molecules only this gel can allow. That's why in this that acts as a molecular sieve. Okay. Through this one, small molecules can move. Assume this is the sieve pose. In this molecule, can this one go through this sieve?
No,
Can? No. Only the small molecules. This molecule's diameter should be less than this diameter, the sieve diameter. Okay, the molecule diameter should be less than the sieve diameter. Then only the molecules can go through the gel. Okay. This is somewhat stain the molecules. That's why the gel we can say the molecular sieve. Okay, until now the gel electrophoresis, which means the migration of the charged particle under the electric field through the gel. That is the gel electrophoresis. Separate the charged molecule under the electric field. Okay. So in here we can separate the DNA, RNA, proteins. DNA, RNA are always negatively charged due to the presence of phosphate group. But in case of the protein, that may be different. The charge amount, the net charge may be different. Maybe the positive or negative, it is depend on the pH. In case of the positively charged molecules or cations, that may be go towards the negatively charged node, that is the cathode. In case of the negatively charged molecules, anion like the DNA, RNA, that will be go towards the positively charged node. The positively charged node may, we can say the anode. In here, the gel may be the agarose, agar, polyacrylamide amide starch. Commonly, we can use the agarose. So in this gel that act as a molecular sieve that only allows smaller diameter molecules only. That's why we can say the molecular sieve. Until now it's clear?
Yes, ma'am. Yes, ma'am.
The principle of gel electrophoresis. The electric field that is provided through the gel matrix. Is it correct?
Yes, ma'am.
Yes, the electric field that is provided through the gel matrix. And negatively charged nucleic acid molecules that is move towards the positive end. The nucleic acid may be the RNA and DNA, which are always negatively charged, that move towards the positive end. The short molecules that move faster than the longer one. You know, in the race, they commonly say the thin people can run faster than the fat persons. Isn't it?
Yes, ma'am.
The less weight people
Yes,
can
ma'am.
move fast. Likewise, in here, the shorter molecule that move faster than the longer one in the gel matrix. These are the principles of gel electrophoresis. It's clear?
Yes, ma'am. Yes, ma'am.
Then agarose gel electrophoresis. In here, the gel, we can take the agarose. That's why that we can say the agarose gel electrophoresis. Commonly, the agarose we can use due to the less expensive. It is the less expensive medium, the agarose. We can take the agarose from the seaweed or red alga. From the red alga, we can extract the agarose. That's why the agarose, that is the natural polymer. Okay, the polymer that have the macromolecules, that is the polymer. In the seaweed, we can extract the agarose. That's why it takes the natural polymer. The agarose that form a gel after heating with the buffer and allowed to cool. First heating, then after that should be allowed to cool. At that stage, we can get the gel or semi-solid medium. Then the pore size may be predetermined by adjusting the concentration of agarose. Okay, the pore size may be predetermined by adjusting the concentration of agarose. First, the agarose. We'll take the agarose. In this agarose, that is available in the market, it is not dissolved in the water, insoluble in water. The agarose that is insoluble in water. It is available in a powder form in the market. Okay, so the agarose powder, we want to make a semi-solid medium, isn't it?
Yes, ma'am.
Ma'am.
Agarose that is in the market in the powder form. We want in the semi-solid medium. But the problem is the agarose that is insoluble in water. That's why the agarose that should be mixed with the hot water. Okay, that make a filaments like this one. Okay. Then after, if you allow to cool this one, that make a cross link. Is the filament. After cooling, that make a cross link. Okay. This is the agarose semi-solid medium formation. In here, after the formation of cross link, you can see the spores. In this spore size, we can change according to the concentration of agarose. Okay, if you add more agarose, which means initially in this structure observe, if you add more agarose, that also make the cross links. So automatically the initial spore size, that is this one. If you add more agarose, so the spore size will be reduced, isn't it?
Yes, ma'am. Yes, ma'am.
Yes.
Yes,
So if
ma'am.
you add more agarose, the concentration of agarose is, if we increase, the spore size will be reduced. Which means, what do you think about that spore size? If the spore size reduced, what do you think about that one? What is impact on this gel electrophores? Is it impact on that one?
Yes, ma'am. We can see small.
Yeah, good. So if the spore size will be reduced, that automatically affect the molecular movements. The smaller molecules only that allow. Okay. So if the concentration increase, that is impact on the spore size. Spore size will be reduced. In this spore size, that also impact on the molecular movements. Only the small molecules can allow through this gel. Okay, this is the agarose gel electrophoresis. Understood?
Yes.
Yes, ma'am.
Okay. So this is the setup of gel electrophoresis. These are the material combined with this instrument, which are the gel tank, casting tray, gel comb, electrical leads, cover, power supply. Please remember these materials. The gel electrophoresis part you should remember. If you remember only upcoming slides, you can understand. Observe this picture. Okay. Shall I move? Please remember these images. Then only you can understand the upcoming slides. Shall I move?
Okay, ma'am.
Okay.
So, what do you think about this part? What is this one? These are the gel electrophoresis parts available in our laboratory. What's that one? Gel tank. Okay. This one? Casting tray. Casting tray. Okay. What's this one? Gel comb.
Gel comb.
Power supply.
Power supply.
Yeah. What's this one?
Electric leads.
Electric leads. This is the red color one, that is the anode. The black color one, that is the cathode. Okay. Anode. Red color one, cathode. Black color one. Okay, this is the gel tank. This is the casting tree. This is the comb, power supply. This is the overall picture of gel electrophoresis. While performing the gel electrophoresis, this is the structure of that. Okay. In here the comb, you can see the different structures are there. Okay. Different structures are there in the comb. In here, how many combs are there? The comb teeth. How many? This one. How many teeth are there in this comb?
5.
5. 1, 2, 3, 4. In here? This one?
3.
3. 1, 2, 3. Here?
3.
3. 1, 2, 3. So in one gel electrophore setup that have the number of combs are there. Okay. The one setup that also have the number of comb. Okay. According to our needs, we can use this comb. Okay, we will discuss later. So the procedure, we will move the procedure of gel electrophoresis. Until now, it's clear.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am. Yes, ma'am.
Okay. The procedure. First, the preparation of DNA sample for running. In here, we will separate the DNA under the agarose gel. Okay, the procedure will be the DNA separation under the agarose gel. The first step, that is the preparation of DNA sample for running. Under that one, isolate the DNA made up into the solution with loading dye. So how do you isolate the DNA? How? DNA isolation? DNA isolation. DNA, that is the double helix structure, isn't it?
Yes, ma'am.
Yes, ma'am.
If you want the small fragment of DNA, you may use the one enzyme. What's the enzyme?
DNA helicase.
DNA helicase.
Yes, ma'am.
Sorry, I don't know about that one, DNA helicase. Sorry? The new words I heard here. Can you repeat that enzyme again? I will check. The person said the enzyme name? Topoid summarize. Is that correct? The person said the enzyme. Topoid summarize.
He said topo isomerization.
Topo high summarize. Okay, I will check later on that one. Okay, commonly we use the restricted endonuclease. Isn't it? I think you heard in the A level. Yes. So in case of the DNA, that is the double helix structure. If you want to get the DNA fragment, you can use the restricted endonuclease. Okay. With the help of restricted endonuclease, you can get the DNA fragments. The fragments means the small pieces of DNA. That is the DNA fragments. So in this way, you can isolate the DNA. Now you have the DNA small pieces. So in that pieces, you should make a solution with the loading dye. Okay, in here the loading dye, that may be the function of loading dye, may be the to give the color. The loading dye function that give the color. Okay, in the gel electrophoresis, in this setup, in here, this is the gel. Okay, in one point, you have okay. In here, this is the gel. In this area, you place the DNA solution. Okay, and this DNA that is moved towards this direction. Okay, this is a small setup of this gel electrophoresis. Okay, across this gel, the DNA can move. That is the basic setup of the gel electrophoresis. So in here, we should make the DNA solution with the loading dye. You know, the nucleic acid or the DNA, we can't see our naked eye. Can we see the nucleic acid? No. So if the nucleic acid move in here, then along this gel, the nucleic acid should be moved. So if it is move or not, we can see. We want to see. That's why in here we apply the coloring agent. That is the loading dye. Okay. In the physical lectures, if in your face expression, we can understood whether it is understood or not. But in the online class, we can't. If you respond only, we can understood whether it is our point is reachable or not. Likewise, in here, the DNA that can't see in our naked eye, so that we can apply the coloring agent. That is the loading dye. Understood?
Yes, ma'am. Yes, ma'am.
The second, yes. So the first step, the preparation of DNA sample for the running. In the overall procedure of the DNA, that is the in here gel, the gel matrix. Okay, in one place you put the DNA sample. The DNA fragments are there in your hand. Okay, DNA fragments. So in one place you put the DNA samples. If you apply the electric field, this DNA can move. Okay, the electric field means that will give the positive and the negative. The electron movements are there. Due to the electron movements that make a positive and negative nodes. So in here the electric field you apply. In case of the DNA that is negatively charged. So due to that one, the negative charged node that is pull the negative charged DNA. That is negative charged DNA that is attracted by the positively charged node. Therefore, in here the movement occur. It's clear? Or shall I repeat again? Okay. So this is the gel. In here, if we apply the power supply, where the electric field forms due to the movement of electron, where positive and negative nodes are occur if we apply the power supply. So the second point, that is the DNA that have the negative charge. So if in this electric field that have the positive and negative nodes, so the DNA negative charge one that is attract to the positive end. In this way, DNA movement occur. That is the overall view of gel electrophoresis. Okay, in here we will discuss one by one procedures. The first step, that is the preparation of DNA sample for running. Okay, in here the DNA sample in your body, that is in the helix, double helix structure. So we want to cut, we want the specific point of the DNA. That's why we cut the DNA helix, double helix structure to get the DNA fragments. That's why we use the restricted endonuclease enzyme. Okay, if we apply the restricted endonuclease enzyme, you have the DNA fragments. In this DNA fragment, that should be mixed with the loading dye. The loading dye function that give the color, because the DNA movement we can't see in our naked eye. That's why we should apply the color. Okay, then after the color movement we can see, isn't it? Yes or no?
Yes, ma'am.
Yes, ma'am.
That's why in here we apply the loading dye. The second step, that is the preparation of agarose TAE gel solution. Under that one, mix agarose powder with buffer TAE. Second, the boiling under the microwave oven 1 to 3 minutes. You know the agarose that is in the powder form, but we want in the semi-solid medium. Therefore, we should make a semi-solid agarose, isn't it?
Yes, ma'am.
So shall we mix the agarose with the water, the normal water? Is it possible?
No, miss.
No, because the agarose that not soluble in the water. That's why we want to boil. With the boiling, at that time we want to add the TAE buffer. TAE means twice. Note it down. TAE means twice acetate EDTA. Okay. We want to make a agarose semi-solid medium. That's why we want to boil the agarose powder under the microwave oven. In that time, add the buffer solution. That buffer solution is the TAE, triacetate EDTA. Sometimes we can use the TBE, TBE also. Trice borate, BORATE, borate EDTA also we can use as the buffer solution. Okay. The buffer. What do you think about that buffer? Did you heard that buffer solution? In case of the buffer solution, that maintain the pH of the medium. Okay, if we add the buffer solution, that can maintain the medium pH. Therefore, the nucleic acid that can move smoothly towards the gel. Okay. What is the function of TAE buffer?
Maintains the medium pH.
So the buffer solution that will be maintain the pH of this medium. Therefore, the nucleic acid or the DNA that can move smoothly towards the gel. That is the function of TAE. So now you have the melted agarose with the TAE solution. The agarose powder that will be mixed with the buffer. In this combination, we should apply into the microwave oven for the boiling. We allow the boiling until the melting. After the melting, we should allow for the cooling of this solution. At that stage, we should pour this mixture into the casting tray. Did you remember the casting tray? Did you remember the structure of the casting tray?
Yes, ma'am.
Yes, ma'am.
Yes. So like this one, no? The casting tray. The both end are not bounded. So if you apply the liquid, what happen? What will happen if you apply the liquid into the casting tray? What will happen? Because the both ends, the top and bottom end, they're not bounded. So what will happen? What will happen?
Flow out.
Yeah, that is flow. So that this both end we should make a boundary. That's why in this both end we should make a boundary by using the sellotape. Okay. Okay.
Okay, ma'am.
Okay. The casting tray, the both end did not have the boundary. Therefore, if we apply the solid nature, now we have the solid nature of the agarose with the buffer. So if you pour the casting tray in that liquid, that automatically flow off. Therefore, we should make a boundary in the casting tray. That's why we use the sellotape to bound the both ends. Before the pouring of the agarose with the buffer, we should place the comb. This comb, like this one. We should place the comb in the casting tray. And this liquid that should be allowed for the solidifying. After the solidifying, we will take out this one gel comb. Okay. So the casting gel pour into the casting tray with the comb and allowed to cool and solidify. In here, this is the casting tray, the both end that is open for the environment. If you apply the liquid nature of the agarose with the buffer, that is automatically flow off. That's why in here we will make a boundary by using the sellotape and place this comb into this area like here. Okay. So above that one, we should place the this casting tray. The casting tray that have the semi-solid gel also with the well. Semi-solid gel with the well that have in the casting tray. The well should be close to the negative electrodes. Okay. So the fifth procedure, that is the loading the gel. Wells are loaded with a DNA sample. DNA ladder is loaded as a reference for size. Okay, the gel where we should place the DNA sample. DNA sample means the DNA fragments and the loading die. We already prepared in the first step. Yes or no? The DNA fragments with the loading die.
Yes, ma'am.
Yes, that sample we should pour into this well. In this well, that should be near to the negative charge. Negative nodes. What's the reason? DNA in the positive end, so the DNA doesn't move. But our need is the DNA should be moved in the gel electrophoresis. Isn't it?
Yes, ma'am. Yes, ma'am.
If we place the well with the DNA in here, DNA can't move. Okay, because the positive end that is attract to the negative DNA. But if our target is the DNA should be moved towards the gel. Therefore, in here we place the negatively charged DNA. If we place the negatively charged DNA in here, the negative electrodes that will be pull the DNA towards the positive end. The negative that is pull the negative charge, this positive end that is attract to the negative charge. Therefore, in here we will place the well. The first one, that is the reference sample of DNA. The first, the well that should have the reference DNA. That's why in here the first well we can see the DNA ladder. The sixth step, that is the positive and negative leads connect to the chamber, turn on the power supply, where the migration of DNA will occur. After all the setup, we will apply the DNA sample in the well, close the lid and supply the power. Okay, that is the gel electrophoresis steps. In here, this well that have the DNA near to the negative electrode that should be moved towards the positive end. When switch on the power supply. In this area, that is the gel tank that have the TAE buffer solution. Okay, understood?
Okay.
Yes,
Okay.
ma'am.
Then the seventh step, that is the visualize the DNA. So if you assume the migration is enough, turn off the power supply. Then that gel that should be transferred into the iridium bromide dye. Then after that, under the UV light, we can see these strips. Like these strips, we can see the orange color strips we can see under the UV light. Okay, that is the gel electrophoresis. Understood?
Yes, ma'am. Yes, ma'am.
Any doubt? Any doubt? So this is the overview of the agarose gel electrophoresis. Okay, first preparation of DNA sample with the loading dye. Then prepare the agarose with the buffer. That's why in here microwave oven used for the mixing, melting the agarose. Then boiled solution that will be poured into the casting tray that should be allowed for the solidifying. At the stage, we should place the gel comb. After the solidifying, take out the gel comb. At that point, we can say the well. Okay, then after, load the DNA sample into the well. The well should be near to the negative electrode. Then switch on the power. After that one, if the movement is enough, we should apply the iridium bromide. Under the UV light, we can observe these strips. We can observe the DNA strips. This is the gel electrophoresis. Okay.
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 So in here the band movement, the longer fragments that move slowly. This is the well. So if you apply the power source, the larger molecules that move slowly, but the smaller molecules that can move fast. Okay, this is the DNA band movement. So any one of you tell the gel electrophoresis process, what do you understood? What do you understood? Gel electrophoresis. Of course, it is little bit tough. Anything do you gather about this gel electrophoresis in this class? Anything?
Yes.
Yes, ma'am.
What are the things you gathered?
What is electrophoresis?
Sorry.
What is gel electrophoresis and how to prepare it and what is proper solution and what solution we are
Okay.
using.
Can you tell the procedure of the gel electrophoresis? Can you?
First we have to prepare the DNA sample.
Okay.
Then mix agarose solution and preparing the agarose solution using the agarose is solid, so we have to make it heat and make the solution. Then casting tray, we have to paste cellotape and cover it, and we have to put the agarose solution. Then put the comb in it too, and after it we solid, remove the comb and place the sample in the casting tray. Then place it in electrophoresis and give power supply, and we can get the stain of the gel.
Okay. Under the UV light.
Okay.
Okay. So until now we will look the agarose gel electrophoresis. With the help of agarose gel electrophoresis, we will separate the DNA. Isn't it?
Yes, ma'am.
Yes, ma'am.
Yes. The first step, we want to prepare the DNA. Therefore, in the human, we have the DNA in the double helix structure. In each and every DNA point that is responsible for the one structures or the function of our body. Isn't it?
Yes, ma'am.
Yes or no? The genes are responsible for our each and every function structures in our body. Isn't it?
Yes, ma'am.
Yes. For that, here we want to get one DNA. Okay, one DNA segment we want. So that's why with the help of endonuclease. Do you want the spelling of this one? Do you want?
No, ma'am.
No, ma'am.
The restricted endonuclease. RE, can you note it down? RESTRICTED. Restricted endonuclease. ENDO. Induce CLEASE. Restricted endonuclease. With the help of restricted endonuclease, you can cut DNA from the double helix structure. Now you have DNA fragments. DNA fragment means the small pieces of DNA. Okay. Now you have. Then after you automatically the negative ion sign here sometimes. So that have the restrict the movement of DNA. Isn't it? Understood or not? What I am saying? Okay. If we didn't apply the TAE buffer, the pH all around the gel may be differ. Okay. So the electron or the negative charged one that have the restricted movement of this along with the gel. That's why we should apply the TAE buffer to maintain the pH along this gel. That is the function of TAE buffer. Sometimes we can use the TBE buffer also. Triacetate EDTA or triborate EDTA we can use as a buffer. Okay. The agarose powder that should be mixed with the TAE buffer solution. And these combination to the microwave oven and provide the heat until this agarose melting. Then take out this mixture from the oven and that should be allowed for the cooling. After some minutes, the casting tray, the gel electrophoresis that have one part casting tray. We should pour this solution into the casting tray. But the problem is that doesn't have any boundary. Therefore, we can bound by using the sellotape in this end and pour into the mixture into the casting tray. At that time, the gel foam we should place in here. And this setup that should be allowed solidifying. After the solidifying, take out this foam. At the place of foam in here, now we can observe the well or the pit-like structures are there. Isn't it? Yes or no?
Yes, ma'am.
Yes. Then at that area, we should put the DNA, this sample. The gel area, we should place the DNA. Okay. Assume this one. Okay. 3 wells are there. In this well, we should place the DNA sample. Assume the one function. The application of gel electrophoresis, we can find the criminals, the forensic use. Okay. The first one application, that is the forensic use, we can use this gel electrophoresis, which means we can identify the criminal. Assume one thief the thief that is stolen the things in one home. Okay, assume. So at that stolen place, the police they get the DNA sample of the thief. Okay, understood what I am saying?
Yes, ma'am.
Sorry, understood?
Yes, ma'am.
Okay, in one area, in one home, murder. Okay, murder. One murder is occurred. Okay, the police they want to identify the criminal. That's why at the spot they collect the DNA sample from the murder from the criminal. Okay, the criminal DNA we can take from the blood, urine, or the touching area also we can get the DNA. Okay, okay,
Okay, ma'am.
okay. So now the police they have the criminal DNA sample. Okay, they have. And that DNA sample, we want to get the DNA by using this restricted endonuclease. So in this sample, we will place this area. That's the reference DNA. So that in this well, we can say the DNA ladder. Okay, understood?
Yes.
Okay. Then the police they suspect two persons. Okay. They don't know, no, they didn't know who are the person do the murder, but they suspect, assume two persons as the criminal, but they want to confirm, isn't it?
Yes.
Yes.
Yes, ma'am.
That's why get the DNA from such two persons. Okay. And one person DNA sampling here, another person DNA sampling here. After that one, in this film, we should place in the negative electrode. After that one, we should apply the power to make the electric field. At that stage, the DNA that is moved towards the positive end. So the larger molecule that move slower, the smaller molecule that can move faster. Okay. So in here, you already placed the loading die. With the help of the loading die, you can see the DNA fragments movement that give the color. So the DNA move like that. Okay, okay, understood?
Yes, ma'am.
Yes. So with the help of the loading die that give the color. Therefore, you can see the DNA movement. So after that one, after completion of this movement, you can take out this gel and apply the iridium bromide. The purpose of iridium bromide is the iridium bromide can bound the DNA. So that give if you apply this gel under the UV light, that also give the color. Iridium bromide that can bound the DNA. So the coloring agent. Okay, that can bound the DNA that give the color. So under the UV light exposure, in here the gel that show the strips, DNA strips. Okay, understood or not?
Understood, ma'am.
Understood. Okay, then so the assume wait. So now you got the image under the UV light. This is the DNA ladder, isn't it? Yes or no?
Yes, ma'am.
Yes, ma'am.
So this is the first criminal DNA sample. Okay. This is the second criminal DNA sample. So, what do you think about this gel electrophoresis? The police can get the criminal, can identify the criminal from this gel electrophoresis.
Yes.
Yes, ma'am.
Yes. So, what do you think about them? We suspected 1 or 2 is the criminal.
Two.
2. Yeah, good. So, each and every person have the specific gene makeup, isn't it? The specific gene makeup each and every person has. So that in the one, this is the DNA get from the murder area. In the DNA fragment that also match with this one, the second suspect, so that the police can identify the second suspect, that is the criminal. He made a murder. Okay, this is the person do the murder. Okay, in this way, we can apply the gel electrophoresis in the forensic use. These are all about the gel electrophoresis. Now it is clear. Sometimes the procedure step by step you difficult to understand. Now it is clear.
Yes, ma'am.
Yes, ma'am.
Okay. If you won't repeat again this one,
No, ma'am.
no. Okay. So then we will move the purpose of gel electrophoresis to get the DNA fingerprint for the forensic use. We discussed that one, forensic use, to identify the criminals. It is the biological evidence in a criminal investigation, the forensic use. Biological evidence. The second application, that is the parent-sibling identification. Sometimes the parents, they lost their child in earlier time. Then after they found. Sometimes you heard some news regarding this one. Did you heard that one? The earlier time, the parents, they lost their children. After the time, some time period, they found the children. But the confirmation by using this gel electrophoresis, they confirm the sibling or the children that is owned by the targeted parent. Did you heard this incident before? In the tsunami? Did you heard? Yes. In the tsunami time, the most of the parents, they lost their children. So after long time ago, they found. Okay, only morphological identification is not confirmation to the incident. So they should do the gel electrophoresis to confirm the child as their own parent. Okay, that is the one application of this gel electrophoresis. So in case of the parents, the father and mother, in our human body, we have 3 pairs of chromosomes, isn't it? Yes or no? Human.
Yes, ma'am.
Yes, ma'am.
Yes, the 23 pairs of chromosomes are available in the Homo sapiens and sapiens. So the 23 chromosome from the father, 23 chromosome from the mother. In here you can see this is the mother chromosome, this is the father DNA. The child 1, child 2, child 3 and child 4. So in here the first child, in this first child that have the father and the related chromosomes, isn't it?
Yes, ma'am.
So in this first First child parents are these persons. What do you think about the second child? It's not possible. Only the mother DNA only that is correlated, similar to this second child. So it is not possible. What do you think about the third child? Possible. Fourth, not same. Yeah, good. So the 23 will be get from the father, 23 chromosome that will be get from the mother. Combination of 23 pairs we have. So if in the gel electrophoresis is performed, both parents and both father and mother DNA only match with the child only, we can tell that one. The child is from these parents. In this way, we can identify. Then the third one is the identify the hereditary diseases. The hereditary diseases mean these diseases that is transmitted through the gene. Okay, that is the hereditary diseases. The thalassemia, sickle cell anemia, some of the hereditary diseases. So if you perform this gel electrophoresis, sometimes you have this structure. So in this example, the thalassemia disease incidence that is responsible for this gene. Okay, assume in this gene that is responsible for the thalassemia. So if the person have this gene, so automatically we can tell this person have this thalassemia, isn't it? Yes or no? Understood or not?
Understood.
So if we perform the gel electrophoresis, we get these strips. In case of the hereditary diseases, the DNA that have the gene, the one gene that is responsible for the hereditary disease like the thalassemia. So if we cut this area and perform in the well here and perform the gel electrophoresis. After the perform, you can get this image. So in this area, DNA that is responsible for the thalassemia. If the person has this DNA, so automatically we can tell the person is affected by the thalassemia. In this way, we can identify the hereditary diseases. Okay?
Okay, ma'am.
Okay. And the fourth one, genome studies. The studies related to genome. That is the genome study. That we can separate DNA. From the separation, we can go for the further studies. That is the genome studies. These are all about the gel electrophoresis. Until now, we will discuss the purpose and the procedures of gel electrophoresis and some principle of that. Do you have any doubts?
No, ma'am.
If you have any doubts, you can ask. Of course, it is very, very difficult, gel electrophoresis. But unfortunately, in the spot exam, we will display these parts. So remember those one, the gel electrophoresis. Any doubts? This is the laboratory images. Okay. We will display this one. And last time we asked this picture in the written exam. Okay. label the images. Last time we asked this one. And each and every sentence is important in your quiz and meet. Okay. Give example. Gel act as dash. So you should fill the blanks. The molecular C. Okay. Understood?
Yes, ma'am.
Okay. If you have any doubts, you can ask. And one person said that enzyme name to make a DNA fragments. Can you put the chat box in the name, the enzyme name? Because I didn't heard the word before. Anybody did you heard that word? I think Topo High summarize like that. Did you study that one before? Students?
No, ma'am.
One person till that one. The person, can you put the chat? Because I don't know the spelling of that enzyme. If you put, I will clarify the next class because I don't know that name before. I'm sorry for that one. Can you put the chat box, the spelling of the enzyme? Can you? Okay, that's all for the day. Do you have any doubts? You can ask. Okay, toposomius. I'm sorry, I searched in the internet. I don't know the word before, but they mentioned that used in the replication of DNA. Okay, I asked the breakdown of the DNA, but I don't know. They mentioned the replication of DNA by using the toposomes. Okay, the person, the 1939 idea.
Yes, ma'am.
Okay, they tell the replication, the enzyme that is used for the replication and transcription.
Okay, okay.
Okay. Okay, the only enzyme we used in the restricted endonuclease. Okay, until now we heard this only from the A level. I don't know if it is used in newly, but toposomes is not suitable for that one. Okay. Okay. If you have any doubts, you can ask.
Ma'am. Thank you, ma'am.
Thank you, ma'am.
You see, after that, we have to remove the sellotape.
Yeah, we want to remove that one. After solidifying, we didn't place that one. We can
Okay.
remove.
Okay.
Okay. Idea.
Yes.
Yeah. If you remove only, wait, I will show. Wait a minute, please. Okay. This area, this is the casting This is the casting tray. This is the gel tank, no?
Yes, ma'am.
Yes, in this casting tray, we should place it in here. Within this one, we have the TAE buffer. TAE buffer to maintain the pH, isn't it?
Yes.
Yes. So under that one, the casting tray that will be the submerged condition in this TAE buffer. So if you have this sellotape in here, the TAE buffer can't move in this area, no? That can't make a pH maintenance.
Yes, ma'am.
Yes. So that we should remove the sellotape.
Okay.
Okay.
Thank you.
Okay.
Loading dye to observe the movement of DNA fragment. And why then add bromide?
Yeah. Under that UV light we want to get the images, no?
Yes, ma'am.
Yes. So that the etherium bromide we use that one. The
But-
etherium bromide that is bound to the DNA. But in here, the loading dye that not bound, that only give the color. Okay. But the etherium bromide that is bound and that is give the color under the UV light. The
Okay.
etherium bromide only responsible for the response to the UV light. That's why we use that one, etherium bromide. Okay.
Okay, ma'am.
Yes. No. Commonly we use the etherium bromide, but the loading dye, TAE buffer, we have the substitute. But the problem, etherium dye only the solution for the to get the images. But that's a harmful, no? That's why you ask, I think. Isn't it? No, I asked the question. Etherium bromide, that is a harmful substance. Substance. Okay. So, if you use the etherium bromide, you should get the protection. Mask the to protect your hand and your face. That give the carcinogenic substances. So we haven't any option, and instead of the etherium bromide only, we can use the etherium bromide under the UV light. While you use the etherium bromide, you make yourself as the protection. Okay. Okay. But you have the option, the loading dye, TAE buffer, you have the substitute. Okay.